do a protein structure analysis. That means we will predict the structure and then we validate the structure. So that is what the two factors included in this protein structure analysis. And one more thing is that we can predict the structure of the protein in different methods. So what are the different methods are available for protein structure prediction, and why we will say protein structure analysis and protein structure prediction. So these are the things we are going to discuss in today's class with this particular topic. So in this protein structure analysis, we are going to see about primary structure analysis of a protein and secondary structure prediction and tertiary structure prediction. In the tertiary structure prediction, there are three different ways you can predict a tertiary structure. That is by means of homology modeling, or by means of fold recognition, or by means of ab initio model. So in that tertiary structure prediction, we will see three different things. And there are tools are also available to predict the quaternary structure for the protein. Then after that, we will see how these protein structures are validated by using different servers. So we will see those things for this particular protein structure analysis topic. So we will take, so if you want to go for this protein structure analysis, we will again have any one of the protein sequence. So now we will go for UniProt. Okay, then we will take caspase protein. Say if it is you are interested in caspase 9, we will type caspase 9 and we will search the UniProt database to get the caspase protein sequence. Now you can see this. These are the protein sequences which are available in the UniProt sequence. Unfortunately, none of the sequence from the human. Anyway, we'll take one of the sequence. Okay, we'll take the first sequence. Okay. Now we go for this particular sequence. This is a caspase 9 homolog sequence, and we will go for this tab, sequence tab. Now this is the sequence which will contain 399 amino acid. So we are going to take this protein sequence and we will do this protein structure analysis with this particular sequence. Okay. So now we will copy this particular sequence. So now we copy the sequence. We go for protein structure analysis. So first we will analyze primary structure of the protein. So in the protein structure analysis, first we will see about primary structure analysis. So for that we will go to the browser and we will say primary structure analysis. Or if you know any tools, for example, there are different tools are available. For example, Protparam is one of the tool, and then Protscale is another tool. So if we know the name, we will give the name of the tool. Otherwise, we'll type only primary structure analysis and it will give the tool. So now I gave primary structure analysis, Protparam and Protscale. So you can see this. These are the two tools for primary structure analysis. Both the tools are available in XPASI. XPASI means it's an expert in protein analysis system. It contains more than 90 tools for protein analysis, and we will see few tools from this XPASI today. You can see this Protscale is one of the tool which is used for primary structure analysis. So now we are going for Protscale tool. So this is the tool page. Here you can see this. We'll paste the protein sequence to analyze the primary structure. So we'll give this primary structure, the sequence. Now we can select any one of these parameters to predict the structural analysis, especially primary structure analysis. So it is default, this hydrophobicity predictions are already selected. We can change any option or we can go for this particular option. So I will submit the sequence and now you can see this is the sequence we are providing it, and it will predict the hydrophobicity of this particular protein. So how this protein, that is the sequence, shows its hydrophobic nature. So hydropathic natures are represented in the graphical representation. So this is how we can predict different properties of the sequence. You can see this, how many the different types of amino acids are available in the sequences, and each amino acids are provided with this hydropath value. So The hydropathic values we can get it from this particular server. So the other tool we saw is this Protparam. It is going to nothing but a parameters, protein parameters. So now again we will submit the sequence and you can compute the parameters. Now you can see this is the submitted sequence. When you come down, you can see this. The number of amino acid we have submitted is 399 and its theoretical PA. So it might be useful for doing some isolation of this protein, isoelectric point focusing, So that can be identified, that is the PA at which the protein might be separated. Can be identified this theoretical PA value and its molecular weight is given, and then how many number of amino acids with different compositions. So what is the percentage of alanine is present in this protein? So we can identify which amino acid is higher composition of amino acid from this particular tool. And you can see here the negativity charge. What is the type of charge molecules present in this protein? Say for example 56 negatively charged residues and 47 positively charged amino acid residues are present. So totally if you see this, the protein might have more negative charges. So this is how we can identify with this type of protein primary structure analysis tool. You can see the number of atoms and molecular weight, molecular formula. So different things you can identify. Even we can identify their prediction of half-life. So what might be the half-life of the protein can be predicted by these tools. Instability index, then aliphatic index. So GRAVI, that is grand average of hydrophobicity, can also predicted by this particular tool. So like this, different properties of the protein sequence can be identified with the help of this particular tool. So this is what primary structure analysis. There are so many tools are available for this primary structure analysis. These are the tools are given as an example for primary structure analysis. Okay.